Hello, my name is Andrew Rouillard, and I am a postdoc in Dr. Avi Mayan's research lab. In this lecture, I'll introduce several data sets that provide information about relationships between drugs, proteins, genes, and phenotypes. The Link's KINOMEscan dataset contains measurements of the inhibitory effect of hundreds of small molecules against hundreds of kinases. This data is useful for discovering off-target effects of small molecules and understanding which kinase signaling networks are perturbed by which small molecules. Chembl and PubChem are other resources that contain bioactivity data for small molecules. The KINOMEscan dataset is structured as a list of small molecule kinase percent inhibition triplets. This information can be used to create a bipartite graph where small molecules belong to one set of nodes, kinases belong to the other set of nodes, and percent inhibition values define the weights of edges connecting the small molecules to kinases. In this case, the weights can simply be defined as the percent inhibition divided by 100. This gives edge weights ranging from 0 to 1, where 1 corresponds to complete inhibition of kinase activity by a small molecule. This heat map is a visualization of a bipartite graph derived from the KINOMEscan data. Kinases label the rows, small molecules label the columns, and the matrix values correspond to edge weights. In this case, the edge weights were thresholded at 0.95 to create a binary matrix. Colored regions of the heat map indicate which small molecules cause 95% or better inhibition of which kinases. We can see that some small molecules have fairly specific activity against only a handful of kinases, whereas other small molecules perturb much of the kinome. This information can be very helpful for understanding the next data set that I will discuss. The Link's L1000 connectivity map contains measurements of gene expression changes following pharmacologic or genetic perturbation of cell lines. It is very useful for learning the system-wide consequences of applying a perturbation to a cell type. It is also useful for learning which perturbations have similar effects on gene expression and which perturbations have opposing effects. The Gene Expression Omnibus is a repository of gene expression datasets that can be mined to collect signatures of differentially expressed genes for many perturbations. By collecting many signatures from GEO, a resource similar to the connectivity map can be created. The Mayan lab has created several collections of signatures by mining GEO. These collections include single gene perturbations, small molecule perturbations, virus perturbations, and disease perturbations. These datasets were created with the help of the Chrome extension GEO2Enricher, which automates much of the process of extracting signatures of differentially expressed genes from GEO. Signatures of differentially expressed genes are computed by collecting several replicate gene expression measurements for a control condition and several replicate measurements for a perturbed condition, and then applying a statistical method to test if the expression levels of any genes were different in the perturbed condition compared to control. This slide shows gene expression data for computing a single signature. Each column corresponds to a single sample, with the control samples on the left and treatment samples on the right. Each row of values reports the expression level of a single gene. This data matrix is the input to a statistical method, such as the characteristic direction, which returns a single column vector that reports the significance of differential expression of each gene. Tens of thousands of such control versus perturbation comparisons can be performed on the Link's L1000 data. This slide shows the end result of performing differential expression analysis on the Link's L1000 data. Each column in this matrix corresponds to a signature of differentially expressed genes for a single small molecule perturbation. Red regions of the heat map indicate genes that are significantly upregulated following a perturbation, and blue regions of the heat map indicate genes that are significantly downregulated. Because of the massive size of the L1000 dataset, what's shown here in this heat map is only 1,000 columns corresponding to 1,000 signatures of differentially expressed genes. The columns of this matrix can be compared to learn which small molecule perturbations have similar effects on gene expression and which have opposing effects. Even more powerful, signatures of differentially expressed genes for different kinds of perturbations from other datasets can be compared to this collection. For example, a disease signature can be compared to the small molecule signatures to learn which small molecules mimic or oppose the gene expression changes seen in the disease. In general, by combining the Lynx L1000 CMAP with other datasets, we can learn quite a lot of biology. For example, with the KINOMEscan data discussed previously, we know both the kinases targeted by a small molecule and the gene expression changes that occur by inhibiting those kinases. The mammalian phenotype ontology is a controlled vocabulary of terms describing the morphology and physiology of mouse organs, tissues, and cells. Each phenotype is annotated with the genotypes of mice that display the phenotype. The MPO contains phenotype annotations for genetic perturbations of thousands of genes, and therefore provides a wealth of information about gene function. The mammalian phenotype ontology dataset is structured as a list of phenotype-genotype pairs. This already places it in the form of an unweighted bipartite graph. This heat map is a visualization of the mammalian phenotype ontology bipartite graph. Genes label the rows, phenotypes label the columns, and colored regions of the heat map indicate the edges, that is, where gene perturbation was significantly associated with a phenotype. From the clustering, we can see that many groups of genes are associated with similar sets of phenotypes, and we might infer that the products of these genes perform similar functions on a molecular level. The datasets discussed in this presentation can be accessed via these links. The Lynx DCIC also provides access via the Harmonizome to process datasets derived from these resources. The processed datasets are provided in standardized matrix, edge list, and set library formats, and all gene identifiers have been mapped to NCBI gene symbols and gene IDs. By mapping identifiers and standardizing data formats, we have taken care of much of the data cleaning work for you, enabling you to jump into analysis of these data right away.
Mm-hmm.